Champagne. As you know, we have a very busy schedule for the next 2 weeks, and I hope you're ready. My main goal with the first couple of slides is to kind of introduce ourselves first, and then we'd like to get to know you a little bit, and then we'll move on with our agenda. Okay? So, let me acknowledge the people who made this possible. So, you know some of the names from the email, so I'd like to thank Professor Stephen Wolfart, who's going to actually talk in a few minutes. You know Marina from the emails, probably you know Julie McCartney, all from Beckman, and Mike May, and Karen Johnson. And you probably got to know Teu and Fred, who are in the back there, and if you have any kind of problems, any personal issues they can solve, including girlfriend-boyfriend issues. We have a number of faculty. If you look at the schedule, basically everybody volunteered their time for this event, so we're very grateful to them. That's Professor Rohit Bhargava from Bioengineering, Logan Yu, Scott Carney, Dan Wasserman, Brian Cunningham from various departments, mostly electrical engineering. We also had very generous people generously volunteering their time with lab tours. So these are TAs that are going to show off their labs. You're going to get to play with their instruments, so that's going to be a lot of fun. So here they are, including people from Beckman, Scott Robinson, Travis Ross, Katarina from Beckman. We're going to show you the microscopy suite and the visualization lab. So we want to give you kind of a round picture of our campus as much as possible, and also get to know, have some hands-on kind of understanding of some of the labs. In case you're wondering how you can spend 2 weeks with $250 fee, the reason is that we have, again, generous support from the Beckman Institute, our College of Engineering, and the National Institute of Health through NIDID, and also through a very supportive local company, ISS. You're going to get to play with their technology also. And there is an entrepreneurial day, probably you noticed it in the program, where we have another few companies, Andor, Eco, ISS, and Optics, who also helped us with the sponsorship. Now, here's where you are coming from. And I'd like to take the next 2 minutes to actually call your names, and maybe you stand up. We don't clap until the end, okay? So we want to go very quickly to get to know all of you. So we are always happy to see that people are coming from various places on the globe. So about quarters from outside the United States. So I think that's very exciting because you get to actually make friends. And I see previous classes of the Biophotonics School who actually get together at conferences now, and there are people that we've met 5 years ago. So I think this is really cool. So I have your names right here. Okay. So first I have Frederic Banville from Quebec. And Deepika Chaturvedi from Indian Institute of Science. Hello. Quentin Davis from Wyoming. Hello. Chris Edwards from around here. Hold it for 2 seconds so everybody can see, okay? Shu Fang from University of Texas. And Rekha Gautam, Indian Institute of Science. I apologize for all the pronunciation issues. Rebecca Gilson from WashU. No. Sikon He from Hong Kong University. Hello. Uriya Hosseini from MIT. Mikhail Kandel from UIC. Hi. Ji-Yoon Kim from University of Michigan. Hi. Justin Klein from UC Davis. Okay. Chi Hao Liu from University of Houston. There you are. Justin Luo from UC Irvine. Hello. Dolon
Yes.
Dolon Chapa, is that?
Yes, exactly.
Okay. Much. call you Jen. Shaokai Meng from Texas A&M. Okay. Viorel Nastase from Romania. Luke Pfizer from here, from UIC. Ramia Raman from Oregon State. Hello. Lee Kyung Ren from Penn State. Okay. Mohammad Sarshar from UC Riverside. Sabina Fresta, sorry, Texas A&M. Shamira Sridharan from UIC. Manmohan Singh from University of Houston. Chen Wu from University of Houston. Their advisor told me that we got all these students from the lab. That is it. Yang Wang from University of Nottingham. Okay. There's no visa issues, I don't think. But Jing Zhang from Nanjing University. So. And Yanghui Zhao from Penn State. Okay. So, welcome. Now we can clap. Very happy to have you here. The jet lag, for those of you who come from overseas, will go away by the time the school is done. Don't worry about it. So, probably you had the curiosity to look through the schedule. Our structure is kind of trying to progressively introduce you the basic principles, try to bring in the first week at least, bring everybody kind of on the same page. So we have a number of tutorials, introductory lectures on optics, methods, biomedicine, and then we really have top-notch presentations from researchers from campus and also from outside on various subjects that we think are hot today, so nanophotonics, quantitative imaging, clinical applications, neuroscience and neuroimaging, and we have an entrepreneurial day that kind of is trying to give you this other facet of what we do, how we understanding better how we can take the technology to the commercialization stage. And we are very excited that at the end of the entrepreneurial day, we're going to have a keynote lecture by the president of Zeiss USA, Dr. James Sharp. And it's going to be a night full of fun. We're going to have a banquet, and we're going to hand out posters, and it'll be great. So usually we have lectures in the morning. We'll have long lunch breaks, so you get to meet everybody and share ideas, and usually in the afternoon we have hands-on labs and tours. So, with this being said, I'd like to now go to our agenda. Our dean could not make it today, but I'm very happy to introduce Professor Stephen Bopart to say a few words on behalf of the imaging at Illinois, who directs for him. So, Steve, please.
My welcome and congratulations to all of you. It's really great that you're here because I think that we're very excited about this field of biophotonics, and you know, for many of you to come halfway around the world, perhaps to be immersed in this this summer school is really a wonderful thing. And you know, I think all of us, faculty and maybe the older generation, wish we would have had something like this when we were in graduate school, because we had to kind of pull things together from many different areas, but what we've tried to do here over the next 2 weeks is really package what we feel is important for you to come away with just a broad overview, fundamentals, applications, and hopefully generating a lot of ideas so you can go back to your own research programs and graduate education and be able to apply some of what you've learned and perhaps go off in many different directions. So I really encourage you to take this opportunity over the next 2 weeks to get to know one another, get to know, the moderators and the people and the speakers as much as you can. Get to know our campus. Hopefully it's nice weather over the next couple weeks, and let it be a very enjoyable experience as well that you can go back and tell many of your colleagues and peers about. This is the 5th year that we've held this, and it just seems to be getting better every time. So we're really glad that you're here. What I wanted to do is to give you a little bit perspective, broader perspective of some of the things that are going on here at the University of Illinois, and particularly around the lines of imaging. You'll see over the next couple of weeks that biophotonics involves a lot of imaging at many different size scales, molecular, cellular, tissue level. But if you even think about imaging from a broader perspective, we can learn a lot. And so I always start with this. This is something that our director for our Biomedical Imaging Center put together. And it's just to say that here at Illinois, we have imaging on the brain. So I don't know if you all recognize this, that this is our alma mater and the center part of campus, which you'll probably get to know. But of course, we want to know and emphasize what we do here at Illinois is not only think about imaging, but we image the thinking about imaging. And we consider imaging and visualization to be quite pervasive in our lives. So if you think about everything that we use or see, and if we want to take data, research data, or relay information, some of the best ways of doing that is through imaging, because we can condense a lot of information in just a visual representation. So when we think about imaging, it's biomedical imaging, but it's also things such as architecture or the arts, or being able to visualize feelings and data. Every figure that you would generate in any paper you write, you can think of it as an image, as a way of trying to relay information. And so the science and the technology and the applications of imaging really play a major part in our lives. And I think we're all used to now looking at images and even streaming video on our cell phones. And this is really the way that we communicate most efficiently. So a couple years ago, we put together on our campus an Imaging at Illinois initiative. And this was really because we wanted to build a campus community around imaging and leverage a lot of the strengths that we have here in imaging science and technology and applications, and really try to bring together people from many different disciplines to think about imaging as a cohesive goal. We can imagine that in astronomy, they use adaptive optics to improve the image quality of faraway stars. It's the same type of techniques and algorithms that are used now in microscopy to improve our imaging through turbid tissue. And that's just one example of many that we can learn a lot from many different imaging partners and fields. So there's a long history of imaging at Illinois. A lot of the early ultrasound bioeffects took place here, was developed here. There's a bioacoustics research lab here in the Beckman Institute that carries on this tradition. In art and design, a lot of the chairs that you may sit on, the Herman Miller chairs, Bill Stump was here in industrial design. We have a very large and major effort in supercomputing. Back in 1986, there was the first, the National Supercomputer Center was established, and today we have Blue Waters, which is a petascale computing facility here on campus. And of course, just fairly recently, the late Paul Oliver, who was a faculty member here, had received the 2003 Nobel Prize in physiology or medicine for his invention of MRI. So long history. And as we went about developing imaging at Illinois, we started thinking about many different areas, like biomedical imaging, remote sensing, imaging in the arts, chemistry of imaging agents, computational imaging, biological imaging, astronomical, materials imaging. There's really a large collection of faculty and researchers across campus that are interested in this. And so over the last couple of years, we've really been driving this and focusing primarily on biomedical and biological imaging. So of those 8 different areas, half of them relate to biomedical and biological. And these are just some examples where different groups on campus have focused on quantitative histology and ultrasound, really all the major imaging modalities. We've had a lot of different imaging workshops to support this, just not only here in Illinois, but regionally and nationally. And in fact, next month, just as this summer school ends, if anyone wants to linger a few more days, we're having a Frontiers in Elastography conference. So bringing together ultrasound, MR, optical, to talk at how we can use those modalities to look at the biomechanical properties of soft tissue. Well, this is just to represent, we've got a lot of different groups across campus involved in imaging, and the message to take away from this is so much of the imaging that you may do in your work, or any of the research that you do, generally is going to involve people from many different disciplines. And so it's important to be able to talk across those boundaries, to think about, if you're an electrical engineer, to think about biology. And that, I think, is also another good reason for this the next 2 weeks to talk to one another, get to know one another, because you all come from very different backgrounds. Well, our motivation for a lot of biomedical, biological imaging is really for human health and to improve that. And one of the things that we like to focus on with optics, of course, is that we can get down to smaller and smaller scales, to get at the cellular, molecular level, because that's where the disease starts. And if we can detect and treat at that level, we perhaps can do a better job at curing these diseases. Well, not only for that early detection, but anytime we're interested in looking at biological or physiological networks in disease, these complex problems, we really are going to rely on imaging across many different scales to look at structure and look at function across all these different scales. And we have to be able to piece all this information together to take really a systems level perspective of how to solve these complex problems. So again, optical imaging, biophotonics really plays an essential role at those cellular, those molecular levels. I just wanted to go through a couple specific examples of research projects. This is by no means comprehensive, but just to give you a flavor of the different types of imaging projects. Professor Mike Insana in bioengineering and a team are looking at trying to piece together these complex biosystems to be able to look at using imaging or diagnostics at many different scales and developing simulations and models to look at how perhaps tumor cells in the breast will develop from molecular changes all the way up to those structural changes. Professor G.K. Liang in electrical engineering is really an algorithms guy, working on developing fast, real-time cardiac magnetic resonance imaging. So being able then to be able to capture beating heart or respiration events and be able to do this in very fast and real time. Brad Sutton and many others are interested in using MRI to understand the brain and in applications for neuroscience, neuroimaging, just neurology. And that involves not only things such as microvascular blood flow or looking at functional changes within the brain, but even imaging animal models. So these, of course, are maybe an order of magnitude difference in scale, but the technologies allow us to look at, say, the mouse brain with just as much detail. Others are beginning to look at how the brain functions in case of injury. So there's a lot of work on traumatic brain injury and how those dynamic responses change the function of the brain. And you'll hear, in fact, later in the summer school, you'll hear from this team here, Professor Fabiani and Bertone, that are using light for diffuse optical tomography, but being able to look at functional changes within the outer cortex. And instead of using functional MRI, they use optical techniques to understand the dynamics and activity within the brain. And you'll hear from them later. Professor Zaburki in bioengineering is using PET and SPEC and micro-CT with targeted contrast agents to look at things such as angiogenesis. So when there's injury, how do those blood vessels reform? And being able to do this is in a rodent model, but with very high resolution. Bill O'Brien and Michael Ozzie in electrical engineering are looking at quantitative ultrasound. So ultrasound may not have the resolution to see the individual cells, but still the amount of information that the backscattered ultrasound contains can allow them to differentiate what types of cells are in there. Are these malignant cells? Is this a tumor that's developing? Is it a benign tumor? and they can. USANA again is a leader in using ultrasound. So in the case of breast cancer, physicians traditionally have always just palpated the human body to detect disease, but now we can use imaging techniques to do that at a much more sensitive scale. Some of my own work, which I'll talk about at a later time in the lecture, involves intraoperative imaging and translating biophotonic applications, primarily OCT here, to clinical applications, and where we can use this for real-time histology. In fact, Professor Volpart, Barney Volpart, my better half, is using imaging to understand stem cells and track these stem cells, and really I think generate some beautiful images, being able to identify where these stem cells are located, what fluorescent labels they have, and what changes in the tissue are occurring. And then finally, Professor Bargava, we'll hear more from him later, but he's in our Department of Bioengineering and using near-infrared chemical imaging to identify cell and tissue types within histological sections. And so this is a way of making histology much more quantitative. So we're here at the Beckman Institute, and I just want to give you a quick glimpse of some of the facilities here. You'll have tours and be able to see some of these firsthand. But our Beckman Institute is really kind of the crown on our campus. We're at the northernmost end of our campus. If you've seen maps, you've probably already walked halfway across campus from your residence hall. But really here at Beckman, we've got many different imaging facilities. We have an imaging technology group that contains a microscopy suite with very high-end microscopes. Also part of that is a visualization lab that has high-end computers and advanced visualization software to be able to visualize the data that is collected from these imaging systems. We have South Campus, we have an Illinois simulation lab. And this is a lab that has what's called immersive technology. So you can walk into this three-dimensional cube or a cave where your data is projected all around you, and you are now immersed in your data, and you can view it from the inside out. That's used for a lot of different applications, including driving simulators, flight simulators, and it's really a neat experience. And we'll have to see if we can get this other tool as well. In the basement of this is our biomedical imaging center. And whereas the microscopy suite was on the small scale, this imaging center is on the larger scale, looking at preclinical animal models and humans, including MRI and also options for PET spec, ultrasound, and optical. So you'll see some of those. And finally, we feel it's important here for outreach. And so a lot of the imaging that we do, because it's so visual, it's a good way to connect with people that are outside our field. So there are several different programs that have connected with the kindergarten through 12th grade groups in the community. One of those is BugScope, which is kind of neat. Kids, in K-12 will find bugs in their yard. They will send them in. We'll mount those on our electron microscopes, and then the classroom can connect via the internet to be able to control the scanning of that scanning electron microscope to visualize those bugs. Also online are entomologists that can answer questions, and kids can really ask specific questions about the bugs that they found. And it's just really neat to be able to see those features at such magnification. But I think that's one way, imaging that can really connect to these other groups. So I just wanted to maybe end and say that imaging in Illinois, we think about imaging all the time here. We've got a large collection of faculty and expertise. If people have questions, I'm happy to answer more questions about imaging at Illinois. But really the work that you're going to do over the next couple of weeks and the presentations for biophotonics have a direct impact and role in imaging. It plays a critical role, like I said, when we're trying to identify and treat disease at the cellular and the molecular level. Optics is the only way that we can do that. So that's why it's such an important part. Well, once again, welcome, and I'm going to be available if people have questions. I'm in and out, but if people have questions, feel free to email me as well. I think my email address is there. So you can the next couple of weeks as well. And thank you.
Well, thank you very much, Steve. It's my pleasure to introduce next Professor Andy Singer, who's not only a faculty in electrical engineering, but also he directs the Technology Entrepreneurial Center. And he's going to tell us a few words about entrepreneurial efforts on our campus. So thank you, Andy.
Thought about a technology that you're working on that you thought might have some commercial application? So the rest of you are working on something that no one's interested in? Okay, well, so maybe by the end of my talk you'll change your mind. It's very important to think about who is interested in what you're doing. Why? Well, because as you're working and coming up with your results, you're trying to answer some questions. Maybe the only person that's interested in what you're doing is you. Maybe that's also, and maybe if you're smart enough and forward-looking enough, then other people will become interested. But really, the challenge in research is to try and answer questions that will have an impact. And that impact may not happen today or tomorrow, it may happen 10 years from now. But identifying why you're looking at what you're looking at and why you're studying what you're studying will help to drive you and help to open up new research vectors. Here at Illinois, we have a number of faculty members that have asked these very questions and have shifted their research in directions that have had commercial impact, not because we're all trying to get rich, but because we're all trying to answer questions and trying to find how we can help change the world. And what I think is a really exciting shift in the way academic research has evolved in the last 10 or 15 years since I've been on this campus is there's been much more of a recognition that faculty members, graduate students, and undergraduate students being involved in technology commercialization is actually a good thing. It's actually exciting to be able to see your research potentially have a positive impact. That might have a positive impact in a research lab, it might have a positive impact in a company, it might have a positive impact in a company that you create. So one of the things that we try to encourage is all of our researchers, again, graduate students, faculty, and undergraduate researchers, to think critically about who could be interested in what you're doing. And then more importantly, go talk to those people. Now, in this room, some of those potential customers of your research might be right here. Right? So I encourage you to talk to one another and find out what each other are doing. And if there's something you're working on, ask these people whether they find what you're doing interesting or what some of their challenges or problems might be. One of the really interesting aspects of the commercialization process is not inventing something and then going out and trying to sell it. That's an old model that fails, it doesn't work. You invent something, you ask someone to give you a whole bunch of money, and you spend all their money trying to figure out why nobody wants what you made. The new way to do this is think about a new technology or an application area, and that's your research. You write some papers, you develop something new. And then you want to go out and do a lot of asking and listening, finding out, I think people in the breast cancer area, or I think people in radiology might be interested in this technology. Then you go to conferences and you just listen. You say, what are some of the challenges you have? It's my understanding that there's a real computational, real-time issues in what you're doing. Is that true? Can you tell me about some of those challenges? Why is it hard? Why is it difficult? What's the cost drivers in the industry? If there was one thing you could change, what would it be? And then when you start to hear the answers to these questions, you say, I know how to solve them. Then you can go back and you can take your research and you can drive it in directions that can solve questions that matter to people. Now, why is this important for you? Well, you probably want to get a job at some point. And if you can take your research and in your presentation, whether it's for a postdoc or for a job interview for a faculty position or a research lab, you can say, I've developed this technology that does X, but I was just at this conference and I talked to this doctor and this doctor in this lab, and they told me they have a critical issue right now that's costing them millions of dollars a year because they can't solve this equation fast enough. And actually, I can do that. Well, all of a sudden, you're worth millions of dollars a year, right? So listening to people and hearing and understanding what those challenges are can drive you into directions of value. You might decide, you know what I have developed in my research lab is actually so valuable that maybe I could start a company, or I could do something around that that could be of commercial interest. That's great. So the next step there is what? Is it you quit school, you start a company? No. Is it you wait till you finish school, then you go out and hang a shingle and start a company? No. You start doing something now, in your research lab. Why? Well, you've got health insurance, you've got a salary, you've got someone that's paying the bills, you've got an opportunity to write grants and get money to direct towards solving a problem. What do you think helps you get a grant funded? That you created a question that you made up and then you think you have an answer to it? Or that you went and talked to a whole bunch of people that found a tremendous need in society, and you have a way to potentially solve it, and it's only going to cost this because you're at a research university or research lab. That's going to get a grant funded. And guess what? You do that research on that grant, you write some papers. Now you're the world's expert in solving this problem that's of great importance to the world. You get a job, you get interviews, you might start a company. There's all kinds of things you can do. So I encourage you, while you're here, talk to one another, listen to one another, find out what some of the challenges are, not in your field, but in fields that are adjacent to yours. And over the next couple of years or months of your research, think about how you can expand and broaden your research vectors to try and solve some problems of interest to people. Think of everybody that you're talking to as a, quote, customer of your ideas. I don't mean customer in the sense that you're going to sell them something, but really, it's a marketplace of ideas. And if you can find ways to find your research of great value to others, not only is it tremendously rewarding personally to know, hey, I'm doing something that other people like and want and need, but it makes you valuable. Right? It gives you the opportunity to then turn that into a financial opportunity or an occupational opportunity. It's no happenstance that most of the faculty members that you will be talking to or listening to during your summer school year have been involved in commercialization activities. Why? Well, because they've done exactly this. They found ways to answer problems that are of tremendous interest to others in the world. And when tremendous interest arises, you can turn that into an opportunity. That might be opportunity in growing your research lab, that might be opportunity in creating a venture. And there's many ways to do that in between. So some of the best drivers of innovation in the world today are you, graduate students and postdocs, that have the ability to spend time thinking and listening carefully and figuring out how to answer some questions. And then you have the opportunity. You're at stages in your life and in your career when your advisor, someone like me who's old, lost his hair, and has kids, I can't go out and go all over the world and start a business today by myself. But if I've got my graduate students that are willing to help me, that are willing to put that time and effort in, then there's lots of opportunity. And in fact, I've got a graduate student who just graduated last week, and we're doing exactly that. Not because we're trying to make a lot of money or trying to get on the Wall Street Journal, because it's really exciting and interesting. And during the last 2 years of his PhD program, he went to conferences and he listened to problems that people were having. He said, I know how to solve that. And why should I write another paper about this algorithm that I can make faster when I can actually write a paper about an algorithm that can solve that problem that's of importance to that industry? So I challenge you all to think through over the next week or 2 weeks here, and then the coming months and the coming years, how can you make a difference? How can you have impact? Again, you don't have to start a business, but you want to think about how you can take what you're doing and turning it into having impact. So I'll be around during the course of these 2 weeks, and if you have ideas, you want to talk about commercialization, I'm happy to come back. And otherwise, good luck, good fortune.
Excellent. Thank you, Andy, very much. Next, we have a few words from Steve Willie with our Office for Technology Management. Steve is one of those people who take our inventions from the lab and actually formalize them to patents and so on. And so he's going to tell you a little bit about kind of the output of our campus activity. Thank you.
So Andy is old, and what am I? I mean, really getting old, losing your hair, turning gray. Trust me, else out there. And so I'm excited about the fact that in this point in your life right now, the fact that you have really, more hair, or it's gray or black at least, you have time to think about stuff under your prof right now. When you're a professor, when you're someone out there, you have so much time on stuff besides your research, you get lost. But right now you can be focused. So definitely, inventing now is the best time of your life. It's really cool. So my office takes care of patenting and licensing your innovations, if you're U of I, of course. And if you're not U of I, if you're part of MIT or Washington or WashU out there, it turns out if people like me on your campus also will take care of your inventions, bring them along, patent them, and license them. What we do is, my office is to work with inventors, like you essentially, and create applications for patents, create them, prosecute them, maintain them, pay for them essentially. And along the way then, working with you, find potential partners out there. You guys know the partners out there. We have 7. There's 7 of me in my office, just 7 of them. There's 2,000 professors in campus here. Professors will know the people out there way better than I will. So working with you folks to figure out who's out there is important for us, to find partners. Partners for sponsor research, for licensing, or whatever. Partners are important out there. We will then negotiate and execute licenses, taking our patent and putting together from your stuff, taking those in and licensing to companies out there, whether it's startups or real companies out there. It starts with your innovations. Very simply, you see a problem. The problem comes from where? And he just said what now? The problem you want to solve comes from where? 10 minutes ago he said what it was.
Necessity.
Okay. So I want to solve, for instance, why it is my hair is curly or not curly. Who cares about that, right? You care about who out there in the world has a real problem that needs to be solved, whether it's in healthcare, imaging, car manufacturing out there. Talk to people out there. India is so right. Find the problems out there that need to be solved. Do that. So take that problem then. When you have a problem then, you figure out, I have an idea. I can fix that. Okay, cool. Ideas, frankly, are cheap. Getting from the idea to end product takes a lot of effort, a lot of sweat out there, a lot of passion out there. Take an idea, you invent something to solve a problem. Cool. An algorithm, a prototype, or whatever. Then what happens? Your funding runs out. That's it. NSF, NIH, there is. It takes that point right there and stops. They don't pay anything for what was there to a product. What do you do then? Well, take yourself. Go outside companies, go to Zeiss, for instance, or startups. Why startups? That can enable actually venture capitalists to come in here and bring money to you. These are not available via the NSF and NIH. So you get the point right now and you go, oh my gosh, this is wonderful stuff out here, but it just stops. Therefore, go here or go here. Startups in many cases bring along further and then go from there to a different company outside. Startups will rarely go from being a startup to being a full-fledged company forever and ever and ever. It happens, but it's not typical. Now, across here, we're doing companies. Stephen Wolpart back there, of course, and his partners, Scott Carney, have a device based on OCT that's wonderful. The problem it solves is a massive problem out there. Breast cancer tumor resections. It turns out, according to Steve Wolpart, when a woman goes for surgery to remove a lump of the breast, 34% of the time they leave there, the margin is still filled with cancer. All right? So you're a woman, you're going for surgery, remove the lump, take tissue away from the margin, go to histology, pathology, wait a few days, they tell you, you know what, we're sorry about this, your margin isn't clean, there's cancer there. You have 2 choices, come on back or wait. What do you want to do? He has an invention right now, FDA approval process, that can take the breast surgeon in the OR, and test right then and there if they need the margin clean or not. It's profound. It's massive. The idea occurred a long time ago. To get from the idea now to reality and saving people's lives is taking years. A lot of money, FDA process, a big team out there. Their passion isn't making money. Making money is a really lousy passion. It fades. You hit roadblocks, you stop. His passion, I know, is looking back someday and saying, that woman is here because of the fact that I help them survive. And make things work. Our company right now is in the middle of FDA approval, going after how many sites we go apart for?
Probably 5 to 8 different sites.
Okay, 5
around the country.
Big places. Okay. And we have Biophotonics, rescue. With QPI to see how living cells work. Remember a long time ago he says, I want to be able to hear how cells grow. Hear how they grow, healthy and non-healthy. That's his passion. His passion wasn't, I'm going to get rich over microscopy. It isn't at all. So here's some examples in the room here right now. Stephen Beaupart and Rescue doing cool things here. Now, by the way, independent of that, Amy Singer, outside the world of optics, they got a company 10 years ago, thereabouts. They got bought by Finisar, doing very well right now. And on number 2, I think, for you. So we have one company here, 2 companies back there, 2 companies here. Not a coincidence. They're doing this thing. They have to do that in many cases. For examples, Argyle, Beaupart, Selden, Cunningham, Braun, are all beginning companies right now in this world essentially. Okay? Just 5 random essentially from the campus. We license this stuff out to Niespec in Germany for nanoholography. That's a cool thing. I'll give it this place, that's cool. Examentia, Bashir Liberton, developed solid-state nanopore technology for doing DNA analysis, given to Dr. Menapore in Britain. Paul Selvin, Bio-nanogenomics, doing DNA analysis with fluorescence technology. And Orbis, from Professor Kevin Kim, is working on a way to actually encapsulate pancreatic islet cells using the same technology as well. So there's a pattern here. You're finding that the most prolific thinking professors here, they tend to take your technology to the point where funding stops and find a way to go past that. And you by the way, besides engineering, cool businesses here. We have a portfolio of native oxide pixels, which is a laser being used in all Datacom around the entire world up there. These are made by the hundreds of millions, just max out there. And so Nick Oloniak, Chantal Sassi invented this 20 years ago. It came to fruition about 10 years after that. It took 10 years to get going one way. And right now, they're about to expire the patents of this thing, it turns out. And therefore, revenues will go from high to 0 in a matter of minutes. We invent new soybean varieties that resist aphids. How boring is that? Not, okay? Not at all. Imagine now, soybeans are really important. Soybean aphids kill about 1/3 of the crops in America, if not for stuff like this. It's really important. Process to convert corn to ethanol, which was really popular 2 years ago, and not so much now for a bunch of reasons. Software that tests energy grid security. Okay? How boring is that? How vulnerable right now is the energy grid? I read that if you take out 9 substations, all it takes to destroy the entire grid in America. 9. That's not a lot of substations to take out. Not certain ones out there. If you do that, then bringing it back up will take weeks, months. Want to kill America? Save your bullets. Ruin the grid. Okay, that'll do it. Therefore, our software can test this stuff. That's really important. We have self-heal coatings that can help warnings this. What if you have tankers carrying oil, and the boats, of course, they can't float away, they rust, they burst open. This is how solid kind of thing. As well as, of course, taking Honda Civics that have pretty paint jobs on America and fixing that. Microplasma lighting. For people I hear about a way of taking microplasmas and making lighting, not so much in tubes or points, but in nice linear arrays. It's really gorgeous. And now, lately, we have a thing called the transistor laser. If you get a chance, walk around campus to see pictures of Nick Oloniak. Nick Oloniak has won every award in the world, I think, besides the Nobel Prize. He invented the LED 50 years ago. Okay? The LED still isn't in this room. Okay? It's in here, I think, but not in this thing. 50 years later, the LED, as good as it is, in terms of lumens per watt consumed, is the best thing in the world, period. 50 years later, it's still coming along. Transistor laser, also a nickel Oloniak, takes a transistor and with it then takes a fourth port, if you will, making the laser either transmitting or receiving it. As he points out in his own way, if they had built blue waters with his technology, it'd be 1/4 the size it is right now and no water coolers. Okay? Walk around campus, see blue waters. It's a massive building. The water tank up there is a water tank there to use low-cost energy at night to cool the water, which then cools the computers. And for I think that 1/3 of the energy on campus goes towards that one box. So he points out, when this thing is finally approved and understood, it's going to take over the world. Kind of like LED is still taking over the world. All these things, of course, were eventually patented and licensed. But besides, of course, the value of patents and licenses out there, as Eddie pointed out, invent things in areas that teachers and other people out there, because they will then do what? Sponsor research, way important here. Lead in papers, lead to more funding, more research, more papers, more funding, more research. It's a wonderful, wonderful spiral. But do something in areas where people want it to be worked in. Don't do writing signatures to you only. Talk to people out there about what's important to them that you can solve. And do that. I think yes. I have that here. I have pamphlets here for starting companies. I'm a contractor. If you want to, pick them up.
Thank you very much. All right, this was excellent. I have to say, I'm learning a few things too. So I think we'll have a break. Let's say a longer break so we get to shake hands a little bit. So let's assemble back at 10:30 and then we'll start with some basic topics. Thank you.
Look up. Yeah.